So in this situation, over here, you have this thing that looks like a transport protein. In fact, it says right there, transport protein. But if you notice up here, out here, presumably the cytosol, you have a low concentration of solutes, right? And you have a higher concentration of solutes inside the cytosol. But we're watching the solute bind to this transport protein and be transported into this area of higher concentration. So that's not diffusion, right? This is not going to happen passively, basically, right? Going from this to over here. So this transport protein has got to use some kind of energy to make this happen, right? You're going to need energy to go from low to high concentration. So this brings us into active transport. And bottom line is we're moving molecules against their gradient. And whatever we're going to happen to do with that gradient, is various. There's various things we can do with that gradient. Okay, so this is active transport. Energy is the big thing here. It's going to be needed to get those carrier proteins to move solutes against their concentration gradient. And we have two basic types we'll talk about here. We're going to break it down into primary active transport, in which the channels themselves are going to use ATP, right? They're going to add a phosphate molecule to this, which is going to energize it, and it's going to help move stuff against its concentration gradient, right? That's how we're expending the energy. And then we're going to see that we're going to build up a concentration gradient through these primary ones, and then we're going to use that concentration gradient to bring stuff in to the cell against its concentration gradient, right? And we'll see, just like those carrier proteins, there's going to be that saturation, right? That could happen right there, right? So, some of the examples we're going to see, the big ones are sodium, and potassium, hydrogen. All these ones are going to be brought out of the cell or into the cell through these active transport. And our major one that we're going to see over and over again is this sodium potassium pump, otherwise known as sodium potassium ATPase pump, which is going to expel sodium, which, against its concentration gradient. Remember, the sodium is high up here, right? And it would diffuse down, but we're pumping up against it. And it's going to bring potassium with that high intracellular concentration into the cell, right? So you're going to maintain this low concentration of sodium inside the cell by pumping it outside the cell, and vice versa for your potassium, right? So it's going to couple these two right here, and it's going to use ATP to do it, right? And so this is a major, the fact that your cell has low sodium concentrations and high potassium, right, is very crucial for many of its functions, right? So your typical body cell, right, it's going to use a lot of the ATP that is generated to maintain this sort of gradient of low sodium, high potassium concentrate in there, right? Remember, the extracellular fluid, the blood, the interstitial fluid was equal, and then you had this differences in the electrolytes here. And this is one way, along with the basic selective permeability of the membrane, that this is happening right here. All right, so, one of the things, how is ATP used in this? Remember, ATP was the triphosphate. You had 3 phosphate groups. And what these phosphate groups do is when you transfer a phosphate group to a protein, it changes the conformation of that protein a little bit, right? It moves it in some sense. It causes it to change its conformation, and it's going to interact with molecules a little different, right? So we say that's giving it energy when you add this phosphate group. So what ATP does is give this high energy bond to a protein, right? It transfers it over, becomes adenine triphosphate to adenine diphosphate by giving it this phosphate. right? So the phosphorylation of this channel right here causes a conformational change. And the sodium, which fits into these little round circles right here and binds to it, which actually stimulated that phosphorylation, are released, right? Just kind of squeeze them out. You can kind of see these round things change and squeeze them out. That's not actually what's happening. I'm just describing what's in the cartoon, but that's the basic idea. You're changing the shape of the protein and you're releasing the sodium against its concentration gradient right here. So once that happens, and this is in this conformation with this phosphate group attached, potassium is going to bind to those spots. It's attracted to those spots. The binding of the potassium releases that phosphate group, and the protein here turns back into this conformation, and that's going, it releases the potassium, and now it's ready to bind more sodium, right? So this is going on all day. You're pumping out sodium, you're bringing in potassium, right? So that's one of the major things in helping to maintain this high sodium concentration outside and low concentration outside, and vice versa inside. And the ATP is basically changing the shape of these and making and causing this to happen, right? So you need energy because you are pushing everything against their concentration, electro concentration gradients. Primary active transport. During the course, right, we're going to see that this is used, quite a bit, right? You have this high concentration of sodium waiting to get into the cell. And you can kind of think of it, right? If you're in a room, there's two rooms or something, it's really hot in the room, and you open up a window all of a sudden, right? What happens, right? You feel cold air. Really, that's the hot air. You have that heat stored. You open up a window and the heat rushes out of the window right here, right? And so by building up all this potential energy over here by the stored potassium or hydrogen ions up here, all you got to do is open up the gates at some point, and you're going to use this flow of ions out that gate, right, to, in this case, run the generator right here, right? So a dam has all this potential energy. You open up the gate right here, all this water is running through, and it's running the generator, right? So that's what your body's doing all over the place, right? It's going to use this potential energy to do sorts of things, to make other things happen, right? So we don't really go over this too much, but if you remember anything from your basic biology class, you learned about the glycolysis and oxidative phosphorylation, the Krebs cycle and all that stuff, right? You're breaking down glucose. You are taking the energy from its bonds. At some step right there, you had all these NADH molecules, high energy. Basically what those were doing was making these proton pumps right here, pump these hydrogen ions on one side of these mitochondrial membranes, right? And so you had this big store of hydrogen ions on one side of the membranes, and then you had this ATP synthase over here that was going to let that hydrogen, just like the water through a dam, right, go through, and that's going to actually what makes all that ATP, right? So you're using the concentration of hydrogen to kind of make all the ATP, right? That's just an example of using these concentration gradient to do some work, right? And then the other thing which will be relevant to this course, that sodium and potassium gradients right here are going to be the main way that we're going to see that cells maintain these potentials, right? You're going to have this negatively charged inside cells, all these sodium ions waiting to get in. And upon some kind of stimulus over here, these sodium channels are going to open. Sodium's going to flood in there. When it does, it's going to cause what's called a depolarization event, right? And that's going to cause this signal of some sort or another. So you're storing a lot of energy, in part due to these pumps right here. And that's one of the main functions of these primary active transport. And then the other one, relevant to today, are the driving of this secondary active transport here. That is, you're using this energy right? This concentration gradient that's made possible by the primary active transport to drive this secondary active transport, right? Bringing things in against its concentration gradient. For all of these, we're again using the sodium concentration gradient, and you'll see, right, you got sodium, 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 right? All coming in. Why? Because remember the gradient, high sodium out here, low sodium in here, right? So if you open up these channels, sodium is going to flow in. In this case, the channel is coupled such that every time, in this case, when sodium comes in, calcium is going to leave. Sodium comes in, hydrogen leaves, right? So they're coupled, right? Or in these cases, sometimes they're all, you have a low concentration of glucose over here for some reason, or amino acid, you want to bring them into the cell. We're also going to couple that sodium influx with a glucose or amino acid, right? But in all cases, the sodium was flowing toward its concentration gradient, and then the other substances were moving against it, whether they're both moving in together like these symporters, or moving in the opposite direction like these antiporters here. I'll give you an example. Here are some of the major ones, right? Sodium, calcium antiporters are ejecting these calcium, right? As sodium flows in right here. And this keeps your intracellular fluid calcium levels, concentrations kept low, right? Same thing with hydrogen. If you want to get rid of hydrogen from the cell, your cell's too acid out here, you want to get rid of them. We're going to use that sodium influx to pump out, to transport hydrogen out, right? And then for these glucose amino acids, right? Remember glucose, well, we'll talk about that in a second, but sometimes here in the extracellular fluid here, glucose was low, but we want to bring it into the cell. And so we'll couple that with sodium, and we'll bring in glucose or amino acids and stuff, right? And we'll see this in these absorptive cells lining the small intestine, as well as in in the kidneys, right? So I'll talk about that in a second, but I just want to point out back here, right? Remember we talked about these glucotransporter molecules that basically just moving down a concentration gradient. This is what's happening in most of your cells. Most of the time, it's not an issue where you have more glucose in here than you have out here, right? Usually you want the cell needs, it uses up all the glucose. It's, bringing in the glucose as fast as it can right here. So it's going down its concentration gradient. But that's in most of your body cells. When you eat stuff and you have glucose in your digested particles in your intestine, right? That concentration might be lower than it is in your body, right? So you're going to actively bring that glucose in with the help of the sodium channel, right? That's absorption of glucose in your intestine. There's also reabsorption when you're making urine filtrate. You're spilling out a bunch of stuff, water that you need to reabsorb, and glucose as well. You want to reabsorb the glucose. So you're going to bring it back into the cells in this point. All right, so these are two different ways of getting these glucose in. And so one thing I want to mention while we're here, I'm going to have a new little symbol right here with this little pin. It means put a pin in this. You don't have to think of it right now, but I want to bring this up later, right? Here are those sodium glucose channels, right? That were actively secondary active, SGL, sodium glucose transporters, right? They're bringing in with the help of the sodium ion concentration in the lumen of your small intestine into the cell. Right? Once it gets into the cell doesn't want the cell, you're absorbing it, you want to put it into the body. You got a high concentration of glucose out here, you just need to add those glucose transporters. And on the other side of the membrane, it lets it out. All right, so here you're actively pumping it in, there you're letting it go out based on its concentration gradient, right? Which must mean here you had low glucose levels, here you had high, here you had low again, right? Same thing in your kidneys. Right? Your kidney tubules, their filtrate is moving through here. You're actively reabsorbing glucose in different regions. And it's all based on the fact that you're, keeping the sodium concentration low in here so that, they're going to come in with glucose. But then within the cell, glucose is high. And so you have these glucose transporters on the other side of the cell that are just moving according to their concentration gradient, right? So what I want you to take note of here is the location of this so-called SGL2, the sodium glucose transporters, and these glucose transporters themselves, right? Where they are, right? Whether it's facing the luminal surface or the basolateral surface, right? So this will be that localization of membrane stuff. All right, but that is for now. We'll get back to that. But, for as far as these membrane proteins that are embedded within the channel, within the lipid bilayer, and are moving stuff in either from their high concentration to low, in the case of passive transport, or in the case of low concentration to high concentration by the use of ATP. And then once you build up this gradient of stuff, right, you can do this secondary active transportation, right? So this is all moving stuff in and out of the cell because we need that stuff and we want to maintain a certain osmolarity. We want to maintain a certain electrolyte concentration and distribution.